Hello everyone. Today in this video we're going to solve the International GCSE Biology Paper 1 from the January 2021 exam series. Paper code 4BI1/1B and 4SD0/1B. This paper has two codes because it is the common paper for the two specification. First specification is Edexcel Pearson IGCSE 9 to 1 scale and 4SD is the Science Double Awards. So let's start. Question number 1. The diagram shows a human sperm cell. Part A. What is the maximum number of X chromosomes found in the nucleus of a sperm cell? So we all know that sperm cells are gamete cells and gamete cells are always haploid, right? They are haploid cells and they always have the 23 chromosomes, not 23 pairs but 23 chromosomes. That means the sperm cells will have one chromosome from each pair. So they have only 23 chromosomes. And we know that the 23rd pair contains the sex chromosome, that is X and Y. And as I have already stated that sperm cells are haploid cells, that means each sperm cell can only contain one of the sex chromosome in them, that is either X or Y. That means each sperm cell will, there is a possibility that they will contain either one Y chromosome or either one X chromosome. So the answer is 1. Then option B is the correct answer for question number 1A. I hope 1A was clear. Moving to 1B. The middle part of the sperm cell contains mitochondria. Explain the function of these mitochondria. So we know that mitochondria is the powerhouse of the cell. Doesn't matter what type of cell it is. It provides the energy for different movement of a species or of that cell. And we know that sperms actually need to travel towards the egg cells. So they need to swim a distance to reach the egg in order to fertilize it. That means these sperm cells actually need to move. And movement requires energy. And this energy is provided by the mitochondria. And the mitochondria provide this energy by carrying out the reaction called respiration. So it has 2 marks. We can write our answer as respiration occurs in the mitochondria to provide energy for the movement of the sperm. With this, our 1B ends. Moving to 1C. The acrosome contains digestive enzymes. Suggest the functions of the acrosome. We know it is already stated that acrosome has digestive enzymes in it, which means that whenever the sperm reaches the egg cell, we already know that egg cell is covered by its own cell membrane. There are also a zona pellucida membrane, and the sperm cell actually need to penetrate the membrane in order to transfer the male chromosome inside the egg cell so that the two nucleuses can fuse together and form a embryo, right? So zygote. So in order to fuse the cell membrane of the egg cell, there are some enzymes needed, and these enzymes are these digestive enzymes inside the acrosome. These digestive enzymes digest the cell membrane of the egg cell in order to transfer the male nucleus inside the female inside the egg cells for the fertilization to occur. So let's write the answer. The digestive enzymes in the acrosome break down the egg cell membrane to allow the male nucleus enter the egg cell for fertilization to occur. With this, our question number 1C ends. Moving to 1D. Describe the route taken by a sperm cell when it enters the woman's body to the site of fertilization of the cell. So it actually wants you to describe our entire route that the sperm will follow before it reaches the egg cell, in order to reach the egg cell. So first it reaches the vagina. At first it reaches the vagina, and then it enters the uterus, and then the oviduct or fallopian tube, so oviduct, and then it reaches the site for fertilization, that is the end of the oviduct where the eggs are present. So we can write it as site of fertilization. So this is the flowchart that the sperm follows in order to reach the egg cells. So let's write it down in words, that the sperm first enters the vagina, then uterus, and lastly in oviduct to reach the site of fertilization. With this, our entire question number 1 ends. I hope it was clear. Moving to question number 2. The diagram shows a food chain from a lake. Question number 1 says, name the primary consumer in this food chain. So primary consumer is basically the one who comes in the second of this entire chain, that is this small fish, because microscopic plants are the producers and the one who consumes on producers are called the primary consumers. So small fish is the primary consumer in this entire food chain. So it will be small fish. Question number B. The microscopic plants convert light energy into chemical energy. Describe the process that converts light energy into chemical energy. We know that this is basically a question asking you to describe what photosynthesis is and how do they carry out photosynthesis. So at first we need to name the process because it's not stated in the question. So we have to first name the process, that is they do this by a process called photosynthesis. Photosynthesis, because they have an organelle named chloroplasts which can absorb light energy to produce starch, right? So because they have an organelle called chloroplast, which absorbs light energy to produce starch. With this, our question 2b ends. Moving to b1 ends. This was b1. Moving to b2. Give 2 reasons why the why some of the energy in the microscopic plants is not transferred to the small fishes. We know that the amount of food the small fish will get, they will not use the entire food, or the amount of food that the microscopic plant microscopic plant eats will not be 100% transferred to the fishes, because some of the energy is used by the plant itself for its own respiration and other body processes. And again, there will be some parts of the microscopic plants that the small fishes will not eat, and some parts of the plants that the microfishes would eat, but still they won't be able to digest it. And even for these two reasons, some energies are still getting lost. So as it has only two reasons, so we can choose any two of the three I have stated. So I'm going to choose go for the options 1 and 2, that is, some energy is lost during plant respiration, and the small fishes cannot digest every parts of the plants. With this, our B2 ends. Moving to question number C. The birds do not have teeth. The fish they eat is initially crushed in part of their digestive system called a gizzard. Suggest why crushing the fish in the gizzard helps the bird to digest the fish. Because whenever we crush some things, it increases the hidden surface area, right? So more surface area are exposed now, which means that the enzymes can now work more effectively to digest the food better. So that's why crushing increases the surface area, so that enzymes can work better on them to digest it. So this is the answer, and let's write it down, that crushing fishes increases the surface area so enzymes can now work on them more effectively. Digest it more effectively. With this, our question 2 ends. I hope question 2 was clear. Moving to question number 3. The diagram shows the human circulation system with some blood vessels labeled A to Z, the A to J. The arrows show the direction of blood flow. So this is the diagram showing the different blood vessels, and the arrows represent the direction of the blood flow. The table below gives a statement about the content of blood vessels in this circulation system. Complete the table by giving the letter of the blood vessel that matches each statement. So this is the statement that we need to match it with the letter of the blood vessels. First one says carbon contains the least carbon dioxide. So contains the least carbon dioxide, it means that it has to be the pulmonary vein. And if we look up into this diagram, here it comes from the heart from the lungs to the heart. So that means A is the pulmonary vein. So option A is the answer for the first blank, so it will be A. Second, it says contains the most glucose after a meal. Contains the most glucose after a meal would be this G, because from gut to liver it connects the liver to gut, and in liver the actually glucose removal and everything happens. So option G is the correct answer. contains the least oxygen, it has to be the vena cava. So it will be J, least, wait, sorry, contains the least oxygen. Yes, it has to be the pulmonary artery, sorry, not the vena cava, pulmonary artery, because from the heart it goes to the lungs. So that's why, in order to get pure, in order to remove all the oxygen, so remove all the carbon dioxide and get more oxygen, so it has the least oxygen present in it. So it will be pulmonary vein, so pulmonary artery. So J is the correct answer. Then contains the least urea, that is F, because here it gets filtered out of the kidneys, and after blood comes out of the kidneys, that means they are filtered and they do not have any urea. So this is the F, contains the least urea. Contains blood at the highest pressure, it is aorta. So blood leaving from the heart to the body, from the heart to the body is B, that's aorta. So B is the correct answer. I hope question number 3A1 was clear. Moving to 3A2. State 2 is in which the structure of blood vessels A differs from the structure of blood vessel J. We know A was pulmonary vein and J was pulmonary artery. Now it doesn't matter what type of pulmonary or not, but at the end of the day, J comes into the criteria for arteries and A comes into the criteria for veins. And veins have always thinner walls, and they have like broad lumens, right? So these are the criteria for veins, and arteries have always thicker walls and narrow lumens. So these are the differences between the vein and arteries. So we can just write the differences from one of the veins or arteries. That is, A has thinner walls than J. Vessel A has thinner walls vessel J, and as A is a vein, so it will have wider lumen. So vessel A has wider lumen than vessel J. With this, our 3A2 ends. Moving to part B. Muscles in the legs contain capillaries. As scientists investigate how long-term training affects the number of capillaries in an athlete's leg muscle, the scientist determines the mean number of capillaries in millimeters square in samples of leg muscle from the athlete before and after a period of training. The table shows the scientist's result. So, the muscle sample before training, and the mean number of capillaries found in the muscle before training is 313, and the mean number of capillaries found in the muscle sample after training was 349. We can see that the number of capillaries increased due to training. Now the question asks us, the scientist concludes that training improves the athletic performance. Discuss the conclusion. We need to discuss the conclusion in detail, and whenever it says discuss the conclusion, we need to go for the both options. We need to both criticize the results, and then we need to again make good points for the points that would go for this investigation, for this investigation, and again against the investigation. We need to both find out the good and bad sides of the investigation. So the conclusion is again partially correct, because after training the number of capillaries increases. We can see that before training the number of capillaries was 313, and after it increased to 349. And as the number of capillaries increases, it means that more oxygen will now be transferred to the leg muscles, and thus they can respire aerobically respire more to provide enough energy for the muscle contraction for the athletic movement. But again, what type of athletic performance are they carrying out is not mentioned. Like it could be either bodybuilding competition or running competition or anything, right? So it's not mentioned that if the athlete is a runner or swimmer. If he is not a runner or swimmer, then his increase in number of capillaries in the leg muscles will not that much benefit his performance, because he is not using his legs, except he is not a running athlete or a swimming athlete. So type of performance is not stated, and there could be other factors affecting the investigation, like age, then lung capacity, then heart rate, and experiment should also be carried on a large sample of persons before we can conclude or come to a conclusion, right? At least there should be 50 to 40, 70 people in an investigation of same age, sex, and same living style, living standard, and then we can conduct an experiment for a long time, and then we can conclude from our experiment. That would reduce random genetic variations also. But just doing an experiment on one person and make conclusion based on that one person is not a relevant answer, is not that much relevant and fair test. So that's why it has both sides. Both one good side is that it increases the number of capillaries that will eventually increase the athlete's performance. But again, if he is not using his legs in the athletic competition, then this will not benefit him in that much different level. And again, it is conducted on only one person, so the investigation should not be considered valid also. So let's write our points. The conclusion is partially correct. Because after training, number of capillaries increase, so more oxygen is transported. To the leg muscles. To respire aerobically to provide enough energy. For more muscle contraction. But the athlete's performance can also be affected by other factors. Such as age, lung capacity, or heart rate. And also the experiment should be repeated on a group of athletes to be reliable. Also the experiment or maybe investigation. No, they didn't say anything, so we can just write experiment. Also, the experiment should be repeated on a group of athletes to be reliable. And with this, our 3B ends. I hope question number 3 was clear enough. Moving to question number 4. 4A, it says the table gives some examples of biological processes. Complete the table by giving the name of each process. The first one has been done for you. Example of process, and we need to name the process from its example. First one is saying that plants with a short growing season survive drought. This is a natural selection. Then it says growth of algae in river pollution by fertilization. We know that when we apply fertilizers on our land, due to raining, the fertilizers will leach off into the rivers, and there, due to high amount of nitrates are present in the fertilizers, they initiate the growth of algae on the surface of the water, and this entire process is called the eutrophication effect. So we will write eutrophication. Pollen transferred from one plant to another by an insect. This is pollination, and as the pollination occurs due to insect, so it will be an insect pollination. Absorption of nitridines from soil using ATP. Like they are using energy to move minerals against the gradient. So obviously this is active transport. Active transport. Part A ends. Moving to part B1. Zinc is poisonous to many plants and can affect natural selection. A scientist investigates the ability of one grass species to survive at different distances from a zinc mine. The scientist uses a sampling method to measure the percentage cover of this grass species at different distances from the zinc mine. The graph shows the scientist result. We can see that the grass species has distance from mine in meters and percentage covered by the grass species. We can see that up to 100 meters from the mine, the grass species has 100% coverage. That means the population for grass is suitable around the 100 meters of the zinc mine, and after that the percentage coverage decreases slightly in almost 105 meters from the zinc mine, and after that the decrease is quite bigger, right? Like from 100% it decreases to what, 20 in almost 180 meters from the mine. The percentage coverage of that grass decreases from 100 to 20%. So these are the readings that we can make from the graph. Let's move to the question. B1 says the zinc concentration in soil is higher near the zinc mine than it is further from the zinc mine. Explain how natural selection could be responsible for the results shown in the graph between 0 and 100 meters. So as we have already analyzed the graph, that during from the distance between 0 and 100 meters from the mine, the percentage covered by that grass species is 100%. That means they have the suitable requirement of everything in this area 100 meter away from the zinc mine. Now this is a natural selection because it was already stated in the first paragraph of question B that most of the plant plants actually cannot tolerate zinc, and they cannot survive in high zinc concentrations. But this grass species has a unique criteria that they can actually survive in high zinc concentration, which means that no other plant can survive in this high zinc concentrated area. That means they will not have to compete for sunlight, they will not have to compete for the space, they will not have to compete for minerals in the soil, and thus they can grow to their maximum rate because they get everything they want, the exact suitable environment for their growth, and that's why they have the 100% coverage around the 100-meter distance from the zinc mine. I hope I have made the points clear, so let's write it down. B1, from 0 to 100-meter distance from the mine, more grass grows because other plants are killed by high zinc concentration. 100-meter, more more grass grows because other plants cannot survive high zinc concentration. So there is less competition for sunlight and minerals. Also, due to mutation, the grasses can survive in high zinc concentration. And thus they can reproduce and pass on the advantageous allele to their offsprings. Pass the advantageous allele to the offsprings. With this, our part B1 ends. Moving to part B2, describe a method the scientists could use to compare the population size of the grass species at 50 meters and 100 meters from the mine. So in order to find the population sample in different areas, we can obviously, the scientists can obviously use a quadrant of approximately 1 meter square, and for both the 50 meters from the mine and 100 meters from the mine, and he need to count the number of grasses in each quadrant, and then he should repeat the experiment 5 more times and calculate the average percentage covered by the grass species. So the scientists can use can randomly place 1 meter square quadrat at 50 meters from the mine and 100 meters from the mine. Count the number of grass in each quadrat in each quadrat. They should repeat the experiment 5 more times calculate percentage covered by the grass species. With this, our question 4b2 ends, and also the entire question 4. I hope it was clear. I'm moving to question number 5. Farmers can keep their animals indoors or outdoors. A student uses this apparatus to compare the heat loss from the animals kept indoors and outdoors. He uses a covered beaker to represent animals indoors and an uncovered beaker to represent animals outdoors. So this is the diagram indicating a beaker with thermometer inside a cupboard, indicating the animals that are kept indoors, and a beaker without any cardboard box, indicating animals that are kept outdoors. So this is the student's method. Pour 200 centimeter cube of hot water into the beaker. Measure the temperature of the water in each beaker. Cover one beaker with a cardboard box. Measure the temperature of the beaker in each beaker. Measure the temperature of the water in each beaker after 30 minutes. The student repeats the investigation 6 times and calculate the mean temperature of the water for each beaker. The student takes the table shows the student's result. So when at the initial temperature, both the initial the temperature for both the beaker is same, that is 80, but after 30 minutes the uncovered beaker loses its temperature by 40 and the covered temperature loses its temperature by 80 minus 44 is 36. So covered beaker loses temperature less and uncovered beaker loses a bigger temperature. So give the dependent variable in this investigation. The dependent variable here is the temperature, because that's what we are measuring. The covered and uncovered is the independent. Like if the animals are kept indoor or outdoors, this is a independent variable. The dependent variable here is the temperature of the cattle that are actually being examined. So the dependent variable here is the temperature. So we're just gonna write temperature. Give one reason why the student uses the same volume of water in each beaker, because volume of water can actually affect the heat loss, right? Because for example, if you took 500 centimeter cube of water and another one has 200 centimeter cube of water, we can easily say that the 200 centimeter cube of water will cool down more quickly than the 500 centimeter cube of water, because 200 centimeter cube of water has less water. So volume is a very important factor because it is directly proportional to the temperature, right? The more the volume, the more time it takes to decrease the temperature. So that's why volume should be kept constant in order to make this entire investigation a fair test. So that's why the volume should be kept constant. So one reason is that because volume affects the heat lost, so it is kept constant. To make it a fair test. Moving to question C, calculate the percentage difference difference between the percentage decrease in temperature for covered beaker and the uncovered beaker, right? So let us first find the percentage decrease in temperature for the uncovered beaker. Percentage decrease in temperature for uncovered beaker and that would be 80 minus 40 over 80 initial temperature into 100%, and if you calculate it you get a value of 50%. And now we need to find the percentage decrease in temperature for covered beaker, and that would be 80 minus 44 over 80 into 100%, and that would be 45%. So with this our question number C ends. I hope 5C was clear. Moving to 5D. The student concludes that it is better for farmers to keep their animals indoors. Discuss the conclusion. Again, as it is saying that discuss this conclusion, it means that we need to point out both the good version of this investigation and also the bad version. We need to go for the investigation, present point, for the investigation, and present point against the investigation. So points that are for the investigation is that if the animals are kept indoors, it reduces their heat loss, but this also again depend on the environment, because if the environment is too hot, even though doesn't matter if they're kept indoors or outdoors, they will have a lot of heat loss. But if the environmental temperature is too cold, then the heat loss won't be that much big in even though they are kept indoors or outdoors. So heat loss of animals actually depends on the environmental factors also. Doesn't matter, it also depends on the obviously the fact that if they are kept indoors or outdoors, but it also depends on the environmental temperature too. So and another point it should be that the difference is quite small. Like from part C we can see that animals that are kept outdoors, they have a 50% decrease in body temperature, and animals that are kept indoors, they have a 45% decrease in their body temperature. The percentage decrease from the two differences is 5%, and 5% is a very small decrease or very small difference. So a very small difference is not always considered to be valid. And again, if they are kept indoors, that means that the animals will not move that much, so they will have more energy for growth, and this also protects them from predators. Like if they are kept outdoors, anyone or anything can come up and eat the animals, but if they are kept indoors, then they are actually getting a protection from their predators. But also the fact that when they are kept indoors, they have to be kept in a very clumsy area, right? The farmers usually do not have much space to keep the animals in a big area, so they have to like fit all the animals inside a small area, and that means a very small disease can easily spread over the entire animal population inside that farm, because there is a crowd build up inside the farm. So a small or simple disease can be very dangerous if they are always kept inside indoors. So these are the all the points for and against the investigation. So I guess I've made my points clear, so I'll just write it down. By keeping the animals indoor, animals indoor causes reduced heat loss, but this also depends on environmental temperature. Temperature and the difference between the decrease in heat loss is very small. Heat loss is very small to be considered valid. Also, if they are kept indoors, they will move less, so more energy for growth, and this protects them from predators. But keeping them indoors can easily but keeping them indoor will make any disease spread easily because of the crowd. Crowd. I hope question number 5D was clear. I'm moving to question number 5E. It says, because the student could modify this investigation, describe how the student could modify this investigation to find out if an animal's body size affects heat loss. Obviously, body size would affect his heat loss, because body size means surface area, and if there is more surface area, more heat loss. If there is less surface area, less heat loss. So as we have to describe how the student could investigate the effect of body size on heat loss, he could use two beakers of different size, and both should be uncovered, so that there is no point for indoors and outdoors. We have to just make sure that the two quantities in order for this investigation are the surface area of the beakers indicating the body size of the animals and the uh, temperature of the beakers indicating the body heat of the animals. So let's write it down. To investigate the effect of body size on heat loss, investigate the effects of body size on heat loss. He could use two beakers of different size and keep the two beakers uncovered for 30 minutes and check the temperature of the two beakers at the initial and end of the investigation using a thermometer. Beakers at the start and end of the investigation. Using thermometers. With this, our question number 5 ends. I hope question number 5 was clear. Moving to question number 6. Neurons transmit nerve impulses. The diagram shows a neuron. This neuron is involved in the withdrawal reflex of a finger from a hot object. The neuron transmits electrical impulses from the central nervous system to the effector. Part 1. Draw a circle in the diagram to show the part of the neuron where the impulse are transmitted to the effector. So we need to find like a circle on the axon terminals, because from there the impulse are transmitted to the effectors, because the axons are connected to the effectors. So it will be a circle over here. Then moving to part A2. What is the name of this type of neuron? We can see that this cell has the cell body at one side of the entire structure. It has a very long axon and it has small dendrites connected to the cell body. So which means that it is option B, I guess, motor neurons. So option B is the correct answer, motor neurons. Part 3 says explain the advantage of a withdrawal reflex when a finger touches a hot object. Okay, the advantage of withdrawal reflex is that we do not need to think before we do our action, do the action, right? If you need to think, then it would be a time consuming, and by that time we would have already hurt ourselves. So to prevent or to cause the at least damage, it is a better option that these actions are involuntary actions. These actions does not require our brain thinking stuff, so which makes it a lot faster, and thus to prevent the highest amount of damage to our body. So let's write down the answer. Withdrawal reflex is a involuntary action. Which makes it a lot fast to cause the least damage. With this, our A3 ends. Moving to B. Some neurons do not have a myelin sheath. Scientists investigate the impulse speed for neurons with different diameters. She does this for neurons with a myelin sheath and neurons without a myelin sheath. The graph represents the scientist's result. So this is the graph. The y-axis represents the speed of impulse, and the x-axis represents the diameter of the neuron in micrometers. And they have, she have investigated two types of neurons: myelinated neuron and non-myelinated neuron. So this type of graph actually give informations about the speed of impulse based on their diameters, not on their myelin or myelinated. So part B1 says which conclusion is supported by the graph. As I've already stated, that this type of graph give information according to the axis labeled. Like this graph will give you information about the speed of impulse based on the diameters of the neuron. So option A says impulses are always faster in neurons with a myelin sheath, which is not correct, because over the entire period we don't know if they it actually have an effect or not, because the x-axis does not provide an label for myelin or non-myelinated. It provides a label for diameters. So option A is not correct answer. B says neurons always have a myelin sheath. Again wrong, because obviously there is some neurons that are non-myelinated. This is shown in the graph, that there are neurons that are also not non-myelinated. So B is also not the correct answer. Wider neurons have myelin sheath, which is completely wrong, because at this point it is a very wide neuron, but they are also non-myelinated. So C is also not the right answer. Wider neurons have faster impulse, which is a very correct answer, because it says that wider neurons have faster impulse. Doesn't matter if they are myelinated or non-myelinated. Both graphs have an increasing effect with increase in diameter. That means speed increases when diameters increases. So option D is the correct answer for B2. B1. B2 says a neuron with a myelin sheath has a diameter of 1 micrometer. Use the graph to determine the speed of an impulse in this neuron. So with a myelin sheath, right? So this is one. We can see this is basically 4.4 meters per second. The speed of the myelinated neuron with diameter of 1 micrometer will be 4.4 meters per second. So impulse speed is 4.4 meters per second. I read this from the graph. I hope it was clear enough for B2. Moving to B3, the neuron is 90 centimeters long. Calculate the time taken for an impulse to travel along this neuron. Give your answer in standard form. At first, we need to convert the 90 centimeters into meters. So 90 centimeters to meter, it will be 0.9 meter, and this is the distance that the impulse need to travel. And we have already we already know from our previous part that the speed of this diameter, impulse speed for this diameter was 4.4 meters per second. So we can use the equation speed is equals to distance over time. We need to find the time taken for this impulse transmission, so it will be 0.9 over 4.4, and if you calculate it, you get a value of 0.20 seconds. As the question stated, you need to give your answer in standard form. That means it will be 20 into 10 to the power negative 1 seconds. I hope B3 was clear. Moving to question number 7. Scientists investigate the effect of salt concentration on the germination of maize seeds. Batches of seeds were watered with different concentration of salt solution. They count the number of seeds that germinate in each batch for 12 days. After showing, they then determine the percentage germination. A1 describe how the students could tell if a maize seed had germinated. We know that germination is a visible change. We can see roots coming out of it, shoots coming out of it, or if the seeds has grown a little or not. So these are like visible changes. So as it has already stated one site or one way that the scientists could tell if the seed has germinated. So we just need to go for the one option, is that they can check if the seed germinated, or they can check they can check if the seed has shoots growing out of it or not. If the seed has shoots growing out of it. Part A1 is done. Moving to part A2. Scientists measured the germination after 12 days. State two other abiotic variables that the scientists needed to control in this investigation. Abiotic means environmental conditions. So as it is related to plant, and plants always have to photosynthesize, and the different factors for photosynthesis are light intensity, then pH of the soil, carbon dioxide concentration, oxygen concentration, and also as this is a investigation between concentration of salt and germination, so volume of salt would also be a factor for this investigation. As it says only two factors, so we can just go for intensity of light and concentration of carbon dioxide. Part A2 is done. The table shows the scientist's result. It has two columns: percentage different germination and the concentration of salt solution. Plot a line graph to show the effect of concentration of salt solution on germination. Use a ruler to join your points with straight lines. Okay, so the percentage germination, at first we need to identify that which heading will be on the x-axis and which heading will be on the y-axis. So we know that always independent variables lie on the x-axis, and concentration of salt solution here is the independent variable because it does not depend on anything. So this is the independent variable, and percentage concentration, this is the factor that depends on the salt concentration solution. So this is the dependent factor. Dependent variables always lie on y-axis and they always lie on x-axis. I have already plotted the graph. The graph would look something like this. Concentration of salt solution in millimoles x-axis and percentage germination on the y-axis, and I have plotted the graph, and the graph looks something like this. And with this our part B1 ends because they wanted to plot the graph. We have already plotted the graph, and labeling the axis is a very must point because it was also stated that we need to label the axis, and it also has 5 marks. So 2 marks are actually for the axis labeling. Moving to part B2, explain the effect of increasing the concentration of salt on germination. From the graph we can see that the line is a sloping downward, and it means that high concentration of salt solution decreases the percentage germination. We need to give explanation for this. The explanation is that solutions containing high salt concentration means that they will have lower water potential. Lower water potential means that the seeds cannot absorb enough water by osmosis, and if they cannot absorb water, then they cannot conduct many of the hydrolytic reactions for their growth or to activate the enzymes to digest starch or to like perform many of the stages of the photosynthesis reaction. So that's why the percentage germination decreases as salt concentration increases. I hope I have made my points clear enough, so I'll just write down the answer. The graph shows increasing salt concentration decreases percentage germination. Germination because high concentration of salt solution has low water potential, thus less water is absorbed by osmosis to activate the enzymes. So with this, our question number 7 ends. I hope it was clear enough for you all to understand. 7C is still incomplete. So let's move to question number 7C. As plants grow, they produce roots and steam. Compare the responses of roots and steam to gravity. So we know that roots grow towards the gravity, like they grow inside the land, so they grow in response to the gravity. But stem, they always grow towards the sun. That means they grow always against the gravity. So these are the 2 points that we need to write about roots and stem. So let's write it down. Roots grow towards gravity. So they show positive gravitrophic effect. And shoots grow, or sorry, not shoots, stems, and stems grow against gravity. So they show negative gravitrophic effects. So with this, our C1 ends. C2 says compare the responses of roots and stems to light. So we now need to compare the responses of roots and steam towards the light. We know that roots grow away from the light, so they show negative phototrophic effect, and stems grow towards the light, so they show positive phototrophic effects. So let's write it down. Stem grow towards the light, so they show positive phototrophic effect, and roots grow against the light, so they show negative phototrophic effect. And with this, our entire question number 7 ends. I hope it was clear. Moving to question number 8. The table shows the data on crops and pesticides used in Norway in 2011. So there are, we can see that the different types of crops, and each crop type has different criteria again, and the total amount of area covered by the crops and the percentage of each type of pesticides, herbicides, fungicides and insecticides. These are the three pesticides used on the each crop. So this, all these things are featured in this graph. Let's move here. It says state what is meant by the term pesticide. We know pesticide is basically a chemical that kills other weeds or unwanted plant. So let's write it down. This is a chemical that kills other pesticides. With this, our 8A ends. Moving to 8B. Determine which crop had the largest area spread with herbicide. Show your working. So we need to find the crop which has the highest area covered with herbicide. So highest area covered with herbicide, we can see that the area amount is quite big for barley, and the percentage of herbicide sprayed on the area was also 91%, which was quite a big number. So obviously barley is the crop on which the herbicide was used over a large area. So let's write it down. The area covered with herbicide on barley crop. So here it will be barley crop. We have already identified the crop and we need to just find the area now. So area covered with herbicide, and that would be 1450 91 percentage. That means 1450 into 0.91, and if you calculate it you get a value of 1319.5. area was measured in kilometers, so 1319.5 kilometers. Kilometers square. Yeah, square. So with this, our part B ends. Moving to part C. Suggest why spring and winter wheat have different percentages of insecticides applied to them. So let's go back to the chart and see for our own self that if the amount of insecticide on spring and winter wheat are different or not. So these are the spring and winter wheat, and the amount of insecticide is 27 on spring wheat and 7 on winter wheat. Now we need to find an explanation that why more insecticide was applied on the spring wheat and less insecticide was applied on the winter wheat. Now the reason is insecticides usually kill other insects, and insects are less available in winter season. Also the insects have less food available, so there will be less insects reproducing. Therefore the insects overall, the population of the insect would be less for winter. That means we don't need to use that much of a big amount of insecticide to just kill the insects, because the population is already less. But in spring the situation is totally different. Spring is a very suitable time for every living creature, and that means for other insects, they will have in spring they will have more foods, so they will reproduce more and their populations would be quite high in springtime. So farmers need to apply a big amount of or a huge amount of insecticides on their lands to protect their plants and destroy the insecticides, high population of insecticides. So that's why the insecticide percentage applied on each area was different. I hope I've made my points clear. So moving to the question to write the answer. In winter, less food is available, so they decrease, and thus so insects decreases and thus require less insecticide. So the insects cannot reproduce that much population. I wrote pollution. So their population would decrease so less insecticide is needed for winter weeds. I hope part C was clear. Moving to part D, discuss the different combination of pesticides applied to fruit and cereal crops. So again, this is a discuss part question. That means we need to write points for the investigation and we need to write points against the entire investigation. So let us first find out the different things that are different between the pesticides applied for fruits and cereal crops. So the pesticides are herbicide, fungicides and insecticide in total. Now we need to compare between the cereals and fruit crops. We can see that there is an even pattern of fungicides in the fruit crops because there is 87 and 80. They are both in the 80s range. So there is an even pattern of fungicide used in fruit crop. But in cereal crops, the pattern is quite widespread. Like the range is from around 620s to 80. So there is a greater variety of fungicides used on cereal crops than in fruit crops. And then we can see that herbicide is used more in herbicides. This is the herbicide. Herbicides are used more in cereals than in fruit crops, and fungicides are used more in fruits than in cereals. Like two of the fruit crops are always getting 80% of fungicides. That means they are providing, or more fungicides are being provided for the food crops. And this is also, there is also a reason because foods are fruits can be easily decayed by other microorganisms or other insects. So that's why they have the high number of fungicides. Also food can decay saprophytically, so also they need high amount of fungicides. So these are the different points that we can see, the differences between the pesticides used in fruit crops and cereal crops. So let's write all these things down. First thing, we saw that there is an even pattern of fungicides for the fruit crops. So let's write it down. There is an even pattern of fruit crops. There is an even pattern of fungicide on fruit crops. On food crops, whereas the pattern of fungicide on cereal crops was a very wider range. So let's write it down, that whereas on cereal crops the amount of fungicide was over a greater range. Fungicide over a greater range or variation. And then we also saw that herbicide was more used on cereal crops, on cereals, than on flowers. Fruits. So herbicides are used more in cereal crops than in fruits, and we also saw that high percentages of fungicides are used on fruits so that they are prevented from saprophytic decay. So let's write it down, that high percentage of fungicide is used on fruits as they are prone to saprophytic decay. So with this, our question number D ends. I hope question number 8D was clear. Moving to question number 8E. Explain an alternative to insecticides that a farmer could use. We know that insecticides are chemical that basically kill the insects that are eating all the crops. So in order to kill or destroy the insects, we can introduce the method for biological control. For example, we can use the predator species that would, or we could introduce, we would release the predator species into the farm, and the predator species will eventually eat all the pests, and eventually there will be no insects to harm the crop. So let's write it down. The farmer can use the concept of biological control use this concept of biological control by using predator or by releasing predator species on his farm to eat the specific pests. And with this, our question number 8E ends, and also the entire question 8. I hope it was clear. Moving to question number 9. Sickle cell anemia is a condition in which some of the person's red blood cells develop abnormally. The diagram shows the red blood cells from a healthy person and red blood cells from a sickle cell anemia. So this is from the healthy person's blood, and this is the blood from the patient with sickle cell anemia. A person with sickle cell anemia often suffers pain as some of their blood vessels become blocked by sickle cells, suggest why the person's blood vessel may become blocked. So the reason is that this sickle cell's anemia always tend to clump with each other. They always end up getting stick with each other. So if the sickle cell red blood cells are going to stick to each other, that means they are going to form a big lump of red blood cell, and if that big lump travels through narrow blood vessels, obviously they will block out the entire blood vessel. The entire big lump of sickle celled red blood cells will block the entire blood vessel, causing to prevent the transport or supply of oxygen, thus eventually resulting in pain or other things, right? Mainly pain. So let's write it down. It's just 1 mark question. So the answer for this should be sickle shaped red blood cells tend to stick with each other. Sickle shaped red blood cells tend to stick to each other. And with this, our 9A1 ends. 9A2 says people with sickle cell anemia have symptoms of tiredness and joint pain that get worse if they are exposed to cold temperatures and high altitudes. Suggest why these symptoms get worse. So first we know that when the temperature is quite lower or when they are at high altitudes, they would reduce the blood flow, and if the cells are already naturally sticking to each other and when the blood flow has now decreased, there is a greater chance for the sickle celled red blood cells to stick to each other now. So the clumping would be obviously increased due to the less temperature and high altitudes. The clumping of red blood cells means that less oxygen is transported to the muscle cells. That means the muscle cells cannot respire enough to produce enough energy, and which leads to the tiredness. Also, the lack of oxygen transportation means that the muscles will now respire anaerobically, and anaerobic respiration causes buildup of lactic acid, and this buildup of lactic acid causes joint pain. So let's write all the points down. Cold temperature and high altitudes reduces blood flow. Reduces blood flow, which increases the sticking of red blood cell, which causes more red blood cells to stick to each other, so which causes red blood cells to stick and transport less oxygen. Therefore, the patient cannot respire to produce enough energy. Which causes tiredness. And also due to lack of oxygen, the patient respire anaerobically, which causes build-up of lactic acid that cause joint pain that cause the joint pain. I also wrote this spelling of respire wrong, so respire. And with this our question 9.2 ends. I hope 9A2 was clear enough. Moving to part B. Here it says sickle cell anemia is caused by a single recessive allele. State what is meant by the term recessive allele. We know that recessive alleles are the ones that can only be expressed in homozygous conditions. So let's write it down. These alleles are only expressed in homozygous conditions. With this our part B1 ends. Moving to part B2. a man and a woman who are both heterozygous for sickle cell anemia, for sickle cell allele have a child, calculate the probability that the child will be female and not have sickle cell anemia. So the child has to be female and should not have the sickle cell anemia. So let us first draw a let us find out that what are their cells types should be, or their own genotypes should be like. So let us imagine that the parent has the genotype of male parent, has this genotype of recessive allele X allele and recessive dominant R, and the female is mother is this. These two alleles are faulty in both parents. Now if we draw a Punnett diagram to find out the probability, that's XR, XR, and that's X R X, sorry, Y R, it will be X R X R, YR. So we already have been told in the first part that sickle cell anemia is a recessive allele. So that means the 4 alleles of the child, let me write it down in here, X R X R, this is the first one, then X R X R, then X capital letter R, Y capital letter R, and then X small r, Y capital letter R. So these are the 4 chances that the child will have this kind of combinations. Now only this combination will lead to sickle cell, will lead to a child with sickle cell anemia, and that is a girl. So we can see that being a girl, there is a 50% chance of being a girl, and the probability of not developing sickle cell anemia is 75%. So let's write it down. We need to find the probability that the child is a female and not sickle cell anemia. So it will be probability female and not have sickle cell anemia, and that will be 0.75, because this stands for 0.25, this stands for 0.25, this stands for 0.25. So not having sickle cell anemia, there is a chance of 0.75% for not having a child with sickle cell anemia. So it will be 0.75, and in order to have the gender of the child a female, there is a 50% chance, so that's why into 0.5, and if you multiply it, you get a product of 0.375. So there is a probability of developing a child who is female and does not have sickle cell anemia is 0.375. I hope entire working for B2 was clear enough. Moving to C. Sickle cell anemia is more common in countries where malaria is found. This is because having an allele for sickle cell anemia can reduce the likelihood of developing malaria. Which type of organism causes malaria? We know they are caused by the protoctist, so option D is the correct answer. Moving to option D, what is the name of the pigment found in the red blood cell? We know that's hemoglobin, so option B is the correct answer. Moving to question number 10. It has question number E. Give 2 differences in structure between red blood cells and white blood cells. We know red blood cells are smaller and white blood cells are bigger. Red blood cells doesn't have nucleus, and red blood cells are biconcave in shape. So let's write all these things down. Red blood cells are smaller than white blood cells, and red blood cells doesn't have nucleus. Does not have nucleus. So with this, our part E ends. Moving to question number 10a. Substances can move into and out of cells by a variety of mechanisms. Explain the factors that affect the rate of movement of substances into and out of cells. So we know that movement of any movement of particles would obviously be related to temperature, because an increase in temperature increases the kinetic energy, which indirectly means that the particles will now move faster. And then an increase in concentration gradient will also increase the rate of movement of particles across the cell membrane. Also, most of the particles actually move across the cell membrane by diffusion. So therefore, if you increase the surface area to volume ratio or decrease the distance, then the rate of diffusion would also increase. And also the size and mass of the particle matters. If the particle is smaller enough and light enough, they can easily move across the cell membrane, and that too in a fast motion. But if the particle is quite big and heavy, that means they obviously cannot pass through the cell membrane, because they will get stuck into the protein carrier chains. And also they it would be very time consuming to diffuse a very heavy particle. So that's why a smaller particle and light particle can move across the cell membrane more easily and in faster motion. So let's write all these points down. So let's start with temperature. The temperature affects the movement and increase in temperature increases the kinetic energy, so particles move more faster. An increase in concentration gradient increases the rate of movement. All the particles move by diffusion. So all the particles move by diffusion. Therefore, decreasing the distance diffusion distance and increasing the surface area to volume ratio would increase the rate of diffusion. Also, smaller and light particles can move faster and easily across the cell membrane. can move easily cell membrane. So with this, our 10A ends. Moving to 10B. Describe the differences between diffusion and active transport. So this is quite easy. We know that diffusion is a passive process where particles move down the concentration gradient, and active transport is a process where energy is used and particles are forced to move against the concentration gradient. Active transport requires carrier proteins, whereas diffusion doesn't require anything. And diffusion can also happen in non living organisms, whereas active transport can never happen in non living organisms because they require energy, and energy can only be produced in living organisms. So let's write all the points here. Diffusion is a passive process where particles move down the concentration gradient. And active transport uses energy. Energy to move particles against the concentration gradient. Active transport requires carrier proteins diffusion. Active transport can only occur in living organisms, whereas diffusion can occur in both living and non-living organism. With this, our question number 10 ends. I hope it was clear. Moving to question number 11. Bread can be produced from flour using yeast. Design investigation to find out if changing the starch content of the flour causes the bread to expand more. Use your answer. Your answer should include experimental details and be written in full sentences. So let's write the experiment. We need to first make 5 dough using same mass of flour, water, and using same species of yeast on every dough. And we just need to change the starch content of each dough so that we can easily identify the effect of starch on the expanding of the flour. And then we need to put the dough at rest for 30 minutes at same temperature, and after 30 minutes we need to measure the height using a ruler. And obviously, as it is an investigation, we should repeat the experiment 3 times for each starch content and then calculate an average. Calculating an average is always a supporting point, is a marking point for this type of investigation questions, because when we calculate an average, it actually makes our investigation more reliable and makes the results more valid. So that's why we should always include the point that repeat the experiment and calculate an average for each starch content or anything that you are going for the investigation. So that's always a marking point. Let's write the answer down. We need to make 5 dough using same mass of flour and water. Flour and water, same species of yeasts are used on each dough. Are used on each dough. The only the starch content is different. Only the starch content is different. We need to keep the doughs at rest for 30 minutes with same temperature. With same temperature, and then after 30 minutes we need to measure the height using a ruler. Using a ruler. Then we need to repeat the experiment 3 more times for each starch content. For each starch content and calculate average. To make this entire investigation, reliable. And with this, our entire question number 11 ends, and also the entire paper for biology, January 2021, paper 1. I hope the entire explanation of this video was clear enough for you all to understand. If you still have any other question that you do not understand, please write it down in the comment section. I would really like to answer your comments. And if you want me to solve any other papers, you can recommend the papers in the comment section. All the screenshots of the answers will be added at the end of the videos, and the official paper and mark scheme, if you want that, you have to email me at mohimaahmedsneha@gmail.com to find the papers. I hope the entire video was useful. Thank you for watching.